So I present about a dynamic-based network for identifying profiling for DNase-Seq. I will first introduce of DNase-Seq, and then I will have protein footprints. Finally, the form definition problem. We know that DNA is order construction. DNA actually is wrapped around the histone proteins of nucleus. And when a DNA region occupied by linear histone is less accessible by transcript factors, transcript factors to bind the free region and regulate generation. Traditionally, DNase-1 has been used to regulate regions, and very recently, DNase-Seq with high-throughput sequencing and to detect the DNA access to nucleotide resolution. And this method actually has been published in our paper. So this technique or digital footprint and step by step. So we first use DNA to digest a population of synchronized yeast cells. And we're back to hard nucleosome-free regions and nucleosome occupied regions, given the digestion is weak. Soft DNA images actually indicates the of DNA. Step is to select the fragments derived from in a close specimen. The ideal region is therefore enriched cleavage. So after high throughput sequencing to our fragments, we map the sequence back and some reads be quickly mapped back to the genome regions. So we figured out the unique mappable, and we finally got this of genome-wide DNA. And so in the next experiment, history means across 80%, which means this 80% have one cleavage. While for the other two unmappable positions, they are mappable positions, but they're actually the unmappable position take about 7% of the whole in the data. So know about its accessibility. We are going to address this from our computational approach. Let's get DNase-1 cleavage tag across. In the following talk, I may use DNase or interchangeably, and they all refer to one cleavage situation. So let's first zoom into a DNase-1 persistent region that enriched for DNase-1 tag. DNase-1 has high avidity and usually, as you can see, this sensitive to the observed regions too. They are depleted of tags, are enriched for tags. So our hypothesis is these two is actually protected tracking factors. Therefore, they were relative to the neighboring regions. For of substance protein binding footprint. So we are giving for each genomic number of cleavage tags, and also whether that position map, and also we are giving the size of our user protein. And then we want a list of non-overlapping fragment statistics to show you how good the footprint. So next time I will present our DBFP based on dynamic-based network. A based hit model. Here I show the network for 2 consecutive positions. Actually, the whole network extends over all genomic positions, and each position is modeled consistently to work. And this position tag counts representing how many cleavage paths observed articulation, and the purple shows whether this position is and the 2 are variable, representing the state of this particular position, and this is what we want to infer from our data. The 2 key points to understand this model is about hidden segment. This segment has 3 footprint, hypersensitive, and background. By assigning one of these genomic position, the DBN actually divides the whole genome into 3 types of segments. Footprint segment, they have background, usually a small length, but the background segments they all have low counts, they have length. And the hypersensitive segments just have high tag counts. And you may notice that there is segment nodes from position select. And this diagram will also sit to transition. Notice addition to the footprint and the background. This is because the segment have to be flagged by segment. Footprints regions of low in middle of high accessibility. The second point is how to handle the map position. Let the variable be a variable tag count. Parent means what is variable? Variable map will take value 2 count were built to the of 6 state, but on the is usually variable or takes false. Therefore, and observed tag count will be in real estate. Basically, the single state of is it to a best face the both sides. Our DBN is a little bit layers of extra to enforce the length count. Not going to details about nodes, but just use to foot segment have length between 8 and 30. We have a length 30. Okay. The next question is how of footprint segment. We are interested in the candidate footprint in router, and we consider a local background window here. If the print contains your tag, the local background window, like what I showed here, this example, will assign deeply go to this print. But on the other hand, if the lump tags in the print at the same even local background, we want to add deletion score to it. So this is also about candidate depletion loss. Basically, have uniform distributed in the back. If a B contains B tag, the probability of footprint containing X or tags actually following by log distribution. Reach A divided by B is the probability of tag into footprint of local background window, and this here is X because measures the probability that the print contains all few. Generally, this probability shows how likely that print is relative to the local background window. So far, we have already seen the first of DBFP, DBN segment the histome and identify footprint segment and the deletion score in footprint. Finally, one to assign TSS score of each footprint. To do this, we shuffle our data in the first two steps shuffled data. This will get critical distribution depletion scores, and then we collect Q each footprint segment. Statistically, discover root, and which a depletion becomes different. Let's describe our digital genomic print data and the footprint data from it. This data is also published in the 2009. The regions span 3 and it covers 2% is one cleavage tax, 3 million map positions with intact mappable positions. The coding which longer than the internet, but their coverage in is one cleavage tax region. These coding regions tends to be and in the J region and stringent Q and cutoff 0.01 we identify 100 difference, and then the cutoff 0 we identify 600 footprints. Similar in coding region, the of 0.01 we identify only 50 footprints. With the cutoff we identify 1,200 footprints. To show our upstream footprint and this plot shows the spatial distribution of relative to the nearest start site, and the different series actually identified in different cutoffs. We observe the footprints are enriched in the region upstream 300 meters, downstream 100 meters. And we cut off more and more stringent from 0.1 to 0.01. We observe a higher than the region. With this plot, we show like the footprint identified and how many of them actually come on this X shows the footprint Q value cutoff varying from 0.1 to 0.05, and the y-axis is which how the footprint containing a null motif. We different sets, magnetic sites, the experiment, and the east observation the zoo motif was derived in vitro body correlation and also these trans-Seq motifs. We could observe that as the value cutoff is more stringent, we could observe a higher fraction of footprint that contain the stringent 1.01, actually 80% of the null motif. Shows motifs that were detected our identified footprint at Q.05. There are 47 source of motif. Either it's the magnetic motif. The column shows you the number of motif or identified footprints, and the column shows you the containing particular motif. Notice that may actually contain this motif. Actually, we want to enrichment of the motif. We calculate the average number of motif occurrences and it generates negative issue of these tubes and use this as our measurement. Ethic is P motif. Its average numbers in footprint about 13 times what we would find in this. There are several motifs they have enrichment. In fact, this function related with chromatin structure, and for each motif stable, they occur frequently in footprints because enrichment are all larger than one. Okay, so we have already seen that enrich on the motif, but still there are quite a few of them do not contain any motif. One of the explanations is some of these footprints actually correspond to non-sequential binding. Imagine blue factor specific, and it may act with some other transcription and correlation. So, purple transcription factor. Consider if this purple transcription factor is binding the DNA, it's not sequence specific. So the foot corresponding to the purple transcription binding will not contain any sequence specific information. To test our hypothesis, we first divide or identify the footprint into two groups. Known footprints means each of them actually come from T, while the others are unknown footprints. Also, we quantitate if footprint's distance to its nearest neighbor is given distance showed. So in this plot, the x-axis is the y-axis representing isolated footprint each group. So at a distance ratio, for example, could observe that 60% of unknown footprint isolated, which means each of the 60% is at least one meter away. It's near. And overall, we find the unknown footprints, more unknown footprints to be isolated than the known footprints. In an unknown footprint to co-occur with some other footprint. Consistent our hypothesis that some unknown footprints are maintained in the sequence. So we find other motifs that are enriched known who are neighbors of the footprints. And we first co-occur M1 with some unknown footprint that M1 may with something of non-specific binding protein expression. And we consist with the fact that MCM1 is a co-occurrence factors. The next one is SCB1 only to bind some of its, but its bind was detected in the microarray. In our data, we see that it co-occurs with some footprint. This suggests that binds weakly in collaboration and correlation expression. Only footprint had for and now we move to the co-ages. In the system need about 287 scripts. But the array is opposite to we actually identified 22 footprints nearing antisense transcribe. And additionally, regulatory for this nearby at the top one. This column is the co-aging where this transcript located nearby the Q value of the between the footprint and the distance of the end. So among the most significant identified from coding region. There are 50 to the shield 6,000 contain and 30,000 contain ABF1, 12,000 contain white. Both ABF and have established control. So our congestion is actually crude open frame and increase the bindability. So in summary, digital footprint divide the nuclear mass across the whole genome. And this type of new, and we develop a new approach DBFP and identify thousands of significant protein yeast in genomic regions. They are enriched upstream and they are enriched no motifs. And we found co-occurrence that contain no motif, which maintained by sequencing. Also, by this, and they are enriched by Chromatin also. Finally, I'd like to thank my advisor, the co-authors on this material use suggestions for her, and our biological collaborators, and thank my Canadian and the supplementary soon online in this link. Thank you for your attention.
Hi. I think there is high resolution analysis of application. Model statistic. Dynamic Bayesian model is a specific and
Strand specific.
No. Yes, in chip analysis, it times we have like a testing problem. If it's solution, this method can have problem.
Chips.
Chips, we have kind of tech because of fragment size.
The resolution is the resolution.
In a second, the tech observation actually from the original source.
That's a different problem because experimental setting determines have resolution. For the data as down, and it's because the put down and also the sequence in the end. So you extend happening side. This kind of data, it's actually measures the accessibility and nucleotide resolution.
Yeah. So when you find or depleted lesions, does it concern that we use some sequencing bias that or some bias? Like, sense if you could use a control sample and find you said depleted on the sample of interest, not control that sort of aware of that. If that's concerns you about using.
In our method paper last year, we actually asked of control how the change and not. You can find that in the supplementary of the nature method.
So that
no, it's not.
I see.
Great talk. What fraction of your coding located footprint close to license transcript?
What fraction?
Yeah, you mentioned that when you talked about the site code regions, you said that some of them were might be the of an antisense transcript, right?
Yeah.
Wondering that most of the occurrences, or that's
Not a small fraction. There's only like 200 antisense transcripts, and we only identified the footprints of 50. And I identified out footprint loser tau, and there's only 2 close to antisense.
so the majority of region-located footprints were not
Oh,
antisense
but for this
at all.
moving part of the print, focus on the middle ones, the motifs, because the experiment designed for in a enriched region. Right? So for the coding region, that is accuracy much lower than the intergenic regions. Look at one.
Thank you.
Thank you.